Unlocking the Mysteries of Emotions, a fascinating dive into our minds. Understanding emotions is more than just a casual endeavor. It's a profound exploration into the essence of our human experience. What are emotions truly, and why do they play such an integral role in our lives? In the simplest terms, emotions are our innate, intangible responses to our surroundings. They emerge instinctively, operating beneath the level of conscious thought. In the early days of humanity, emotions served a crucial function. Their ability to swiftly alert our ancestors to dangers in their environment allowed them to navigate a perilous world effectively. Fast forward to today, and while our world has drastically changed, the fundamental nature of our emotions remains strikingly constant. However, despite their vital role in our survival, scientists find themselves on a continuous quest to unravel how these emotions are generated within our brains. In a remarkable study emphasized in Science, researchers have recently made significant strides in mapping the brain-wide activity patterns that trigger emotions. Interestingly, their findings reveal a fascinating phenomenon. Once an emotion is activated, it persists long after the initial trigger has faded. This sensation may resonate with many of us. Think of the experience when you accidentally stub your toe, feel the pain surge, and yet remain aware of that pain even after you've pulled your foot away. Or when indulging in your favorite ice cream flavor, enjoyment lingers beyond the last spoonful. Decoding the emotional responses hidden in our blinking. This groundbreaking study, spearheaded by Carl Dieseroth at Stanford University, delves into how emotions manifest in response to unpleasant, yet not painful, sensory stimuli. Participants underwent a unique experiment involving an eye puff assay. A device blew gentle bursts of air into their left eye in precise sequences. Each puff lasted around 60 milliseconds, and to prevent anticipatory reflexes, gaps between puffs varied from 3 to 8 seconds across a 5-minute session. Meanwhile, a high-speed camera captured the participants' eye movements and their behavioral and subjective reactions. As anticipated, the consistent air puffs elicited reflexive blinking. Participants instinctively withdrew from the puffing device. They also exhibited prolonged eye closure or rapid blinking in between puffs, reporting the experience as both unpleasant and annoying. To delve deeper, the researchers included a separate group of participants who were inpatients at Stanford University Hospital and had been equipped with electrodes in their brains to monitor epileptic seizures. Those who gave their consent experienced the same eye puff assay, yielding comparable behavioral responses. Reflexive blinking and prolonged eye closures were evident once again. However, the true intrigue lie within the brain activity that unfolded during this study. The role of ketamine in emotion processing. Every air puff sent signals shooting across the brain, akin to a breaking news alert, succeeded by a prolonged and subtler signal. Through data collected from the electrodes monitoring the participants, the scientists identified specific circuits in the brain that were engaged in the generation of emotional responses. To validate these outcomes, they administered ketamine to some participants before repeating the eye puff experiment. Ketamine is an FDA-approved anesthetic and, at reduced dosages, serves as an antidepressant that can induce temporary dissociation, altering one's subjective experiences for a brief span. This allowed researchers to differentiate between reflexive responses and the emotional ones that followed. Remarkably, the use of ketamine had no impact on the participants' initial reflex reactions or the immediate burst of neural activity. What changed was that the slower brain response dissipated more quickly for those on ketamine. Consequently, participants reported a shift from annoyance to simply perceiving the experience as a tickling of the eyeball. This adjustment in perception correspondingly influenced their behavior. Rather than blinking or closing their eyes, they maintained open eyes despite anticipating more puffs ahead. Ketamine is known to inhibit a sensor in the brain responsible for integrating varying signals. Thus, For individuals under the influence of ketamine, their brains failed to consolidate these signals into a coherent emotional response. From mice to men, unraveling emotional circuits. Despite the vast differences in size and complexity among vertebrate brains, the fundamental blueprint of the brain remains consistent. To further pinpoint the systems that govern emotions, ones that have withstood the test of evolution, Dicerov's team extended their experiments to lab mice. These mice underwent the same eye puff assay. Their brain activity was captured through surgically implanted electrodes, and they received ketamine injections. The team observed similar patterns in the mice as they did in humans. Ketamine significantly altered the firing rates of certain neurons, while others remained unaffected. Only the neurons involved in the second phase, what we now understand as the slower emotional response, registered changes due to ketamine, mirroring the human findings. In this expanded mouse model, scientists could analyze the neural activity following each puff in even greater detail. They discovered a rapid increase in activity following the eye puff in various midbrain regions, including the thalamus, which serves as the nexus for incoming sensory data before it is relayed onward. Also noted was the periaqueductal gray region, which plays a role in emotional behaviors. The second phase of neural activity corresponded with engagement in the brain's emotional centers, often encompassed under the broad term limbic regions, as well as the frontal cortex, precisely in line with expectations. The researchers then dissected the brain activity patterns into distinct stages, scrutinizing the speed at which neuronal activity escalated after the eye puff and how long it persisted. They uncovered a curious finding. Neural activity in various regions spiked sharply right after the puff, but the rate at which it diminished varied significantly. For instance, the midbrain regions displayed a swift decline, whereas activity in the frontal cortex lingered longer. Unsurprisingly, the thalamus remained active during both the initial and secondary phases, presumably due to its role as the central hub for sensory signals, a coordinating function that is what allows for communication across different timescales. Extending the conversation on emotional processing, this recent study is the first of its kind to highlight such differential activity patterns across various brain regions concerning emotions. The implications of these findings on the cortical and midbrain areas remain undetermined. Using computational models that track neuron firing dynamics in lab mice, alongside their behavioral outcomes in the iPuff test, the research team postulated that the timescale of neural activity plays a crucial part in shaping emotional responses. Essentially, if sensory signals dissipate before the brain has the opportunity to integrate the information, it undermines a person's ability to learn from that experience, meaning they might not truly grasp, for instance, that they should shield themselves from an irritating stimulus. Conversely, if brain activity remains overly attuned to the observed fast and slow phases, this prolonged activity can also lead to its own challenges, as overextending brain states are associated with mental health issues such as depression, obsessive-compulsive disorder, and post-traumatic stress disorder, condition causing individuals to grapple with persistent or mismatched thoughts and emotions. Beyond simply labeling responses as healthy or unhealthy, the fast and slow phases of brain activity illuminated within the study could unveil the fundamental mechanics of how our brains process information. Importantly, individuals differ significantly in how they interpret signals from their environment, something that is influenced by both genetics and inherent life experiences during formative years. What if emotions were non-existent? Why do we experience emotions? A more enlightening approach might be to flip the inquiry. What chaos would ensue if we lacked these essential responses? Without instinctive reactions provided by emotions, our brains would rely entirely on slower risk-benefit analyses, a process that could be incredibly laborious. Over time, simple decisions, like what to wear each day, what to have for dinner, what subjects to study, or even whom to marry, would become exhausting exercises in indecision without emotional guidance. This groundbreaking study has opened a window into the brain's inner workings during the initial moments of emotional emergence. As research continues to evolve, it is our hope that forthcoming studies will decode the specific information embedded in this primal circuitry, providing greater clarity on how various emotions are encoded and how they adapt over time.